Alright, so continuing on, let's have a look at how you can interpret a mass spectrum now that you have seen how MS works, okay, the fundamentals and the basic operation of it. So your mass spectrum is, of course, okay, this is the mass spectrum, okay, so you get lots of spectra, okay, so one is called spectrum, okay, a lot of them called spectra. So you have, okay, basically x-axis and y-axis. So x-axis is basically your mass over ion charge, okay, whether sometimes it's m over z or from TOF if you are using MALDI. So the y-axis is usually heat on the detector or number of ions detected, okay, in percentage of intensity. So each peak represents a peptide from your cleavage. Now, a peptide doesn't mean that, okay, it comes from different protein. It may come from different protein. It may come from same protein as well, okay, but it only says, okay, here that, and the spectrum, okay, basically represent, okay, one peak per peptide, not per protein. Now, the nuts and bolts of using PMF methods for molecular weight searching, okay, or basically mouse. So the search is basically based on match my peptide masses. So when searching, coincidental or erroneous matches will also occur, which is quite common. Okay? Even when you do BLAST, did you ever wonder how come they could sometimes give you something which is totally unrelated or from different organism and so forth? So database searches generate lists of candidates, most of which are coincidental matches. So your mission, okay, and this is mostly especially true, okay, when you are searching, okay, for or when you are verifying of a peptide sequence, is to select the best candidate and to justify a claim that this candidate is correct, because it's actually much more tedious as compared to your molecular or your general nucleic acid. So, the mouse search, how to rank the candidates. So it's actually the simplest method that have been used and usually being applied to a lot of researchers. So the candidate that generates the highest number of peptide matches, okay, is of course the top candidate. So you will build a list with the most matches to the least matches, and some engines, okay, usually use this method. Actually, a lot of engines use this method. It's just that they have different algorithm. So it is actually still useful, and of course, again, it's an important character. So a better method is to acknowledge that some peptide sizes are rare. Now, a better mouse, okay, or molecular weight searching, okay, is when a protein or population of proteins is digested and the distribution of sizes of proteolytic peptide product is not random. If it is random, that is actually quite common, so it has been known, okay, in the database. Now, basically, the odds of a match resulting from a simple coincidence are lower if the peptide size is rare and higher if the peptide size is common. So some matches deserve extra impact in your scoring scheme, especially those which are unique or novel. So the problem is, of course, or should I say challenge? Well, challenge is more, of course, probably not as bad as problem sounds. So the database, because they are usually huge and there are too many data in them. So in gene bank alone, okay, we know there are lots and lots of sequence, millions of sequence, probably trillions. Okay? So we know that, okay, data, okay, have been gathered, okay, since 1980s. Okay, as you can see, this is actually the beginning of it. Okay, and picking up, okay, in the mid 90s, and this is basically up until 2013, but up until 2020, okay, we know that the number have increased. Now, what I meant by, okay, the database are huge, okay, is that, okay, because as years go by, okay, you will get new records being uploaded, okay, by researcher into the database. Now, what are the criteria for the upload? Usually it has to be verified, okay, by a study, and usually whatever that is being uploaded, they will be checked, but chances are, even if there is something wrong with it, okay, they will just keep it in the database, probably not to be used as a high matching score, but they will keep it nonetheless. Okay, so this is a repositories as compared to new records. Now, for protein work, and actually a lot of other works, okay, sometimes you have experimental, sometimes you have hypothetical, but in a lot of protein work, usually you have experimental and hypothetical. Hypothetical meaning that you do some prediction in silico studies. So, yeah, this is where, okay, what you did in the wet lab, okay, which is experimental, okay, needs, okay, or basically could be matched, okay, with the dry lab, okay, which is hypothetical, or computer or bioinformatics. So let's have a look at them side by side, okay, so you can actually differentiate them, okay, by the color. So on your left hand side, okay, is actually for the experimental protein. So usually we digest experimental protein with protease, okay, there are different types of proteases. So the size resultant, okay, is actually peptides, okay, and basically they are quite similar, right? So what you have is that you will generate a list of peak masses, aka peptide mass fingerprint, as compared to hypothetical proteins in bioinformatics work or in silico study, whereby you have archive of protein sequences and you have index, okay, which is done via in silico digestion with proteases. So you can calculate theoretical masses of individual peptides and you can generate a lookup table for use by scoring algorithm to allow ranking of the matches. So basically this is done depending on the type of tools they use. So at this point in the technology, the database is not built from experimental mass spec data. Usually it is from hypothetical proteins. Now, how do you interpret again tandem mass proteomics result? Let's have a look. Now, first and foremost is the organization. So basically, what we're going to take a look at how you identify the protein, okay, with tandem mass spectrometry, and then we'll move on the development of some of the bioinformatics technique, okay, and basically I'm going to basically talk about sequence, and probably we'll have a look at X tandem and mascot, which is the most common being used as well. So we're going to take a look at how these programs differ, and basically we can see how we could take advantage of them because they actually or basically are using different algorithm and the scoring is also different. Now, first and foremost, you start with the protein. So your protein is of course again going to be digested first, okay, before you can send it for tandem mass spectrometry for the identification of the protein. So once you have digested and cleave it, so of course hopefully you can get many of them. And well, by what he said as very quickly will actually depend. If you do it by bioinformatics, yeah, it will be very quickly. If you were to do it in the wet lab, it will take some time because there are processes that needs to be done. Now, let's take a look, okay? So if you cut with an enzyme, and the most common enzyme, okay, and we are talking about mass spectrometry, okay, so usually mass spectrometry, the proteases or the common protease, okay, to be used is trypsin, okay, the other one is chymotrypsin. There are other types of proteases that could be used, okay, but usually it's trypsin. So trypsin basically only have two cutting sites, okay, which is at the arginine and lysine, okay, R and K. So we have 20 amino acids, 20 basic amino acids again, and they have different codes. So basically, okay, this technique, okay, basically is kind of like lysing of your protein. Okay? So sometimes you can get about 8 to 20 amino acids in length, but basically it depends on the location of the cutting sites because sometimes miscleavage could also occur, okay, which we will talk in a while. Now, I'm going to give you guys okay, an example of how the results of same protein, and this is of course done in silico, when you cleave it with three different proteases with three different cutting sites. So, well, in this video, I'm just going to show you, but when we meet, okay, in online class, okay, I will show you. So, get ready with your laptop to, well, go along with me. Now, if you want to cleave it with the trypsin, so this is the protein sequence. Now, okay, you know that trypsin basically cut, okay, at the arginine and lysine. So at every R, okay, and K, okay, usually it will be cut. So sometimes you can get very long sequence, sometimes you can get short sequence, okay, even only one. Now let's compare it with chymotrypsin. So chymotrypsin is a derivative of trypsin. So again, okay, take a look at similar sequence, how it is cut. Chymotrypsin cut at phenylalanine, tryptophan, valine, leucine. I actually forgot why it's okay, but you guys can have a look okay, over the internet. So you can see that there are lots of peptides okay resulted okay from cleaving with chymotrypsin, and you actually get a lot more shorter peptides as compared to when you basically cut with trypsin. So let's take a look at the third protease, which is pepsin. Again, similar sequence. So pepsin basically has four cutting sites, okay, which is phenylalanine, tryptophan, again why I actually could remember, and also methionine. So now, okay, you can see that okay, some of them actually only just have one of the amino acids. Okay, some of them are very long. Can you see this? Okay, up until here is actually very long. This is more than 30, I think. Okay, so now my question for you, okay, and this is for you to think of, and if you basically are interested, you can raise a question, okay, when we go online. Um, which is actually preferred, short peptide or longer peptide? And basically, which is better? Now, I mentioned about miscleavage now. Yes, so why are there miscleavage? Now, I'm going to show you again an example of miscleavage. Well, basically, because I'm so used to seeing okay with trypsin, so basically, I'm going to show you okay, the example in trypsin. Now, can you see that what happens here? Okay, supposedly it cuts over here, right? Okay, so this is basically what I'm showing you what is being done. Okay, using in silico digestion. We basically use an online digest tool. So miscleavage could occur sometimes. Okay, by right it should cut here, but it's not cutting there. Okay, and I think for this one that would be the only one that I saw. Okay, if there is any other, then you can let me know. Oh, another one here. Okay, so miscleavage occurs, and it occurs basically quite frequently. Okay, so there are a few causes. For example, non-specific cleavage. So this occurs basically if in the lab, okay, in the wet lab, okay, if you do this, okay, usually it's because of you don't have enough ratio, okay, of the enzyme to the protease. Sorry, enzyme, okay, to your protein. And in in silico studies, okay, so usually this is where you put in the command how many miscleavage that you want to have. Okay, so non-specific meaning that if it is too close by, as you can see before that, okay, so if it is too close by, okay, it could just be missed. So non-specific, creating a non-specific cleavage. Second, the presence of proline at C terminal. So you can have a look back, okay, at well, again, okay, let's have a look again at trypsin, okay, if there is any proline towards D, but we actually don't see it. Okay, so the presence of proline, okay, at the C terminal, okay, which is basically towards the end, sometimes could also cause miscleavage. Why? Proline is one of the, do you guys remember what proline is in the 20 amino acids? It's one of the special amino acid, right? Okay, so go and check okay, what is so special about proline and why it is causing the miscleavage. Again, okay, the third one, okay, the third cause is two successive basic residues, which is basically sometimes could also cause okay, miscleavage. But this is usually only, not to say only, okay, the occurrence is quite mostly seen in wet lab. And the last one is actually the presence of negatively charged amino acid at either the N or the C terminal site. Now let's move on, okay, to the peptides, okay, that we have seen earlier, and we know that this is cut with trypsin, okay, which have the lysine and arginine cutting sites. So what happens is that we look at each peptide individually and we select the peptide by mass using the first half of the tandem mass spectrometer. So this is the peptide that we're going to take a look, okay, in the box. Now, what happens, okay, in the mass spec, okay, and remember back in the mass spec video. So the first step, not to say the first step, probably is the second, okay, so it imparts energy in the collision cell. So this is what happens, okay, so it imparts energy into the peptide, causing it to be fragmentized at the peptide bond, okay, between amino acids. So it basically will cause some loosening, okay, of the bonds. So basically then what happens is that okay, we measure the mass of the daughter ions. So this is recorded using the second mass spectrometer, and this is because we're talking about MSMS or tandem mass. Now, what you have okay from or the results of the tandem mass, okay, or actually just single mass, okay, and basically most of the time it's actually tandem mass, you get three types of ion being produced. The first one is B-type ions. So these ions are basically based on nomenclature, and we are not going to discuss on that because it's actually very complex and I don't really have time okay, to discuss on that because it's actually very lengthy. So there is a mass difference okay, between the peaks, okay, as you can see okay, with each other, okay. Basically, of course, this is directly correlated with the amino acid sequence. Now, if you have a look, okay, at over here, okay, A to E, the distance, okay, minus the A, okay, from here to here, if you minus here, okay, should produce the mass of E, okay, if from here to here, okay, and you minus A or and E, okay, should produce, okay, the mass of P, and so forth. So you can build these mass differences up and of course derive a sequence for the original peptide. So this is, well, to some people it's actually kind of like cool, okay, and it makes the tandem mass spectrometry one of the best tools. But it sometimes okay requires some precision and some targeting. Now, B ions okay usually have a tendency to degrade and lose carbon monoxide producing, which is quite common, and we shall see again the occurrences here on. Apart from B-type ions, we also have A types of ions. Okay, so I actually forgot to mention that B type of ions okay, is actually being read from your left to right, similar with A type from your left to right. So the correlation between A and B ions okay, is that how are they different? Okay, can you see that the location? Oh, sorry. Here. Okay, so basically, okay, the masses is being measured for B type. Okay, throughout the whole of the amino acid, okay, but for the A type, okay, it's actually up until almost 3/4 of it, okay. This is because of the carbon dioxide, sorry, carbon monoxide. So the correlation between A and B ions, okay, is that, oh, okay, so as you can see, okay, the distance, okay, between them is usually around 28 unit, okay, so it's always almost about, okay, 28 units, and you usually don't really see that much of A, but if you do see, okay, and if you were to measure the distance okay, between A and B of the similar ions, okay, they basically have a difference of 28 unit, and the unit is actually being measured in a complicated formula, so we're not going to discuss over there. Now, the third type of ions, okay, is called the Y type. So basically, not to make your life difficult, but probably it is, okay. So it is being read, okay, the other way around, okay, or backwards, okay, from your right to your left. Kind of like, okay, for some of you mostly, okay, if you were to read Jawi, it's the other way around. So yeah, this is Y type of ions. Now, all the peaks, okay, usually have different measured heights, and many peaks can often be missing because it's the other way around. So usually we look at B ions as compared to Y, but sometimes okay, Y types of ions is crucial as well. So then we have it okay, in your typical spectra. You have B type, A type, and Y type of ions. And to make your life simpler, basically you can see that it is color coded. But okay, what we don't even know okay is that okay which type of ions they are okay in real life, because in real life okay what you get off of the machine okay is just black and white such as this. So how do you know which is A type ion, B type ions, or Y type of ions? Okay, so well this of course will make your life difficult, but what can you do about it? Okay, so apart from that there are also noise. Now if you remember, I'm not sure whether it is taught or being told okay, to you, in sequencing, your general nucleotide sequencing, okay, you can get noise. And I'm not sure whether Dr. Fatah covered it for the genomic part. So when you get noise, okay, it's actually a similar thing in proteomics. Okay, so when you get noise, so it'll make your life difficult because you don't know whether the noise okay, is actually the actual peak corresponding to amino acids to your peptide sequence or not. Okay, and okay, combination of them okay, in real life okay, looks like this. No color coded. It's actually black and white. So it is actually quite difficult okay, for you to get to justify or to know which one is Y, which one is A, which one is B, and which one are noises. Now, basically, okay, this could be done okay, in some ways. Okay? And of course, okay, with the help of a computer animated, automated way. So, I will explain further on how it is going to be done okay, in the second video or in the next video. So go and check it out.